Thank you, everybody, for sticking around. This has been a fabulous 4 days. Huge kudos to the Radiopharmaceutical Track Subcommittee for all the people that worked so hard to present. And it is my honor to organize the last session here. Dr. Yasa Kapala is our moderator. He almost needs no introduction among the researchers in radiopharmaceutical therapy. He is program director within the Division of Cancer Treatment and Diagnosis in Radiation Research Programs at the National Cancer Institute. Our first speaker today is Dr. Chris Tikacek. We were just talking about correct pronunciation before the term, and I almost messed that one up. He is a board-certified therapy medical physicist. He is an associate member with the Division of Radiation Oncology at the University of South Florida and Moffitt Cancer Center. Go ahead.
Okay, let me see how to make this work here. Okay, so thanks for that introduction. Happy to be here, and thank you for coming to the last session of the very last day. So I appreciate you coming. Hopefully you will take away some information that you can take to your radiopharmaceutical program. So first off, I'm coming from Moffitt Cancer Center. I had the privilege of working under Dr. Dave Morse, who is kind of the leader of our radiopharmaceutical development program. I did a lot of this work in my graduate studies at USF and Moffitt, and I'm going to kind of take you on a journey through how we came up with this idea for this therapy and how we got it into the first-in-human phase 1 clinical trial, which is active currently. We developed a targeted alpha therapy that's targeted to metastatic uveal melanoma. The trial started, we injected our first patient in late 2023. So it was very exciting for me, kind of seeing the growth of this project all the way through all the mice trials and the in vitro studies, and then finally injecting it into a human. Words can't really express the emotions and feelings that I had on that day. So I hope you take something away from this. So I'll just briefly talk about the funding here. Okay, so and these are in order of how we got the funding throughout the development pipeline. So we got a Bankhead coli pre-spore, that's a Florida grant mechanism. We got a NIH SBIR phase 1 with Intazyme. Then the Melanoma Research Alliance Team Science Award was a multi-year, multi-million-dollar that funded my PhD, so thank you for that. Then we got a SBIR phase 1 and 2, which we teamed up with Modulation Therapeutics, and we actually licensed our IP out to them. So the phase 2 SBIR is how we're funding the trial. Then I got the Melanoma Research Alliance Young Investigator Award to develop, to continue developing this therapy. And then we got another multi-year, multi-million-dollar DoD Melanoma Research Program Award. So I'm sure everybody has seen this picture. Whenever I'm trying to explain the power of radiopharmaceuticals, I always show this picture. This is lutetium-177 PSMA and with gallium-68 PET imaging prior and follow-up. And you can just see the red is, in general, gone across these 8 patients. So a lot of people don't understand what radiopharm is, really. My therapy physics colleagues really have no idea. So I just showed them this, and then they seem very amazed. So this was displayed several times this week, so I'm not going to go too much in this, but this group put together this wheel of current status of radiopharm therapies and trials, and it's just really neat to look at, like, the booming interest in radiopharm therapies. So there, right there, that I highlighted, that's Modulation Therapeutics, that's us. And we are currently in the phase 1, and over here we're using actinium-225, and we're using a peptide ligand. So I'm sure you've all seen this before. This is from the NIH website, kind of the anatomy of a radiopharm therapy. So there's the cancer cell. Then on the cell surface, you have a target protein. Then you have the targeting moiety, which we have been and are going to continue doing peptide ligands. Then you have the linker, and then what's not shown here is the chelator, and then you have the radioactive payload. So for us, this is the schematic of our radiopharm therapy targeted to metastatic uveal melanoma. Here is the targeting peptide ligand. I don't know if you can see my mouse. You can't see it.
So you gotta drag it up, maybe up.
Whoops. Oh, okay, down there. Okay, so this is our targeting moiety. We're targeting the melanocortin-1 receptor, which is expressed in metastatic uveal melanoma. Here is what they call the linker, and here is the chelator, which is the DOTA, and here's our payload. So this is the schematic of our therapy. So I'm going to share with you the pipeline that we at Moffitt have been following to develop these therapies. We have one that's in phase 1, and we have 8 more in the pipeline at various stages. So stay tuned, because there's a lot of exciting development coming in the future. So first, you need to find if you're following this pipeline, we were looking for cell surface target discovery. Then targeting moiety discovery, and we are going with peptide ligands. Then conjugate development. Then radiochemistry optimization. Then preclinical and IND. IND is investigational new drug, enabling studies. So this will be in vitro and in vivo preclinical studies. And finally, the first in human phase 1 clinical trial. So as a physicist, we are really involved in the last 3, or at least in my experience. The first 3 are very heavy in chemistry and biology. But really, when I came on board, we were at this phase right here, radiochem optimization, and then I'm still involved in the phase 1 trial. So cell surface target discovery. This right here, I think, is one of the most important parts of the pipeline. You need to determine the clinical need. And to do that, you need to talk and consult and discuss with clinicians that are working with patients in the clinic. They're interested in collaborating on therapies for a specific clinical indication. There's been dozens and dozens of groups that have not consulted with clinicians and went ahead and tried to make a therapy which will never see the clinic. So if you take nothing away from this talk, make sure you discuss with a clinician that there's a clinical need for your idea. Then you should conduct mRNA expression profiling of data from relevant patient specimens to identify cell surface targets that differentiate tumor from normal tissues of concern for toxicity using a library of cell surface genes. Identified targets are ranked by breadth of expression among tumor specimens and expression level in tumor relative to normal tissues of concern for toxicity. And finally, protein expression is confirmed by patient specimens by immunohistochemistry of tissue microarray or proteomics. So the clinical need. Uveal melanoma is the most common ocular malignancy. Metastatic uveal melanoma is basically a death sentence. It has a median survival of less than 1 year. So there's not even a 5 or 3 year survival rate. So there's no effective therapies currently. It tends to metastasize. I think it's like 85%, 80 to 85% of metastases go to the liver. uveal melanoma has different driver mutations than cutaneous melanoma, so it kind of differentiates itself from cutaneous. So novel targeted therapies with low systemic toxicities are needed for metastatic uveal melanoma. So we had this idea, and we went and discussed with clinicians. So Dr. Kuchelani is the chair of cutaneous oncology at Moffitt Cancer Center, and he is the PI of the phase 1 trial. We also discussed with Dr. El Haddad, who is IR and nuke med physician, and then Dr. Gage, who is a nuke med physician scientist. So the goal, we're going to develop a targeted alpha therapy for metastatic uveal melanoma with low systemic toxicity. So we have an established clinical need. So our target, what are we going to target? So the melanocortin-1 receptor, MC1R, is a member of 5 G protein coupled melanocortin receptors, 3 of which are expressed in tissues of concern. And those tissues of concern are clearance tissues like liver and kidney. So we excluded those and honed in on MC1R. It's expressed in 95% of metastatic uveal melanoma cells, and it's not expressed in primary melanoma, nor is it expressed in healthy tissues. Okay. So MC1R is a bona fide metastatic uveal melanoma target. So we have the clinical need and we have the target identified. So now we need to see what is the expression of the target on cells. So we did an MC1R mRNA analysis, expression analysis, and we found that the MEL270 and the OMM1 and OMM2.3 all have good expression. Then we did confocal micrograph staining imaging. So we did the DAPI, which is the nuclear stain in blue. Then we did the MC1R stain, which you can see is on the cell surface, which is good for us. Then we did the membrane stain, which is in red. And we determined that there are 410,000 MC1R receptors per cell. So this is a bona fide target for us. Okay, so we have the clinical need and we identified the target. Now we need to make the peptide ligand. Now this is pretty heavy in chemistry. So first you want to see if anybody has figured out a structure-activity relationship for promising targets. So you review the literature. If there is a relationship available, you work with a peptide chemist to improve the physiochemical properties by synthesis and testing of rationally designed compound libraries. So in the process of this, we incorporate metal chelator into this process, which include lanthanum and europium, which have similar chemical charges as actinium, but non-radioactive. So here is a busy slide, but I just want to tell you that we did competitive binding assays with europium, where the actinium-225 would be, and MC1RL is what we're calling the peptide ligand, has excellent binding affinity and selectivity for MC1R. So on the table on the left is a library of 8 different ligands that our chemist had developed, with the first one being kind of like a template for the rest. And based on the studies, we went with number 4, and I'm not even going to try to say that. And then in the middle there, where 4 is, you can see the binding affinity and selectivity. So where MC1R column is, compared to MC4R and 5R, we have the lower Ki constant. So when you're looking at binding affinity, you want a low Ki constant. So 0.24 is significantly less than. So MC1R with this compound is is our lead ligand. And then here's some binding plots, and then we publish that. All right, now conjugate development. So in this process, you choose your radionuclide, you develop the conjugation chemistry. So you need to consult with or team up with a radiochemist. Chelation chemistry development, medicinal chemistry, conjugate characterizations, like structure, binding, plasma stability, and cellular uptake. And then in this part of the pipeline is where you can start thinking about companion imaging tracer development. So we went with actinium-225. We obtained it from Oak Ridge National Lab. It has a long 10-day half-life that allows for centralized production, complex decay chain with several alpha-emitting daughters, and it's easily chelated with DOTA, which is the chelator that we went with. So in the conjugate development, we did some studies on the linker optimization, and we tested several different linkers. And if you can recall the schematic that I showed in the beginning, so here's our targeting ligand, and then here are the linkers that we tested out. Here's the DOTA, and after putting the actinium-225 in, we have this. So we did several different linker combinations, and we published that, but we also did 2 other ones in addition on a different publication. After this study, we ended up determining that our lead linker is going to be the aminohexanoyl, AHX. And I think I have some biodistribution graphs that say why we didn't want to go with the other ones. So radiochemistry optimization, in this part of the pipeline, the production protocol by the radiochemists needs to be developed, radiochemical quality control and optimization, and that includes the purity, the yield, the specific activity, plasma stability. You need to have a GMP, good manufacturing practices, SOP, because this is clinical trial, so you need GMP certification. You need to do at least 3 trial runs showing that you can do this correctly. And then again, this is where your companion imaging tracer further is developed, if you're going to develop that. So the radiochemical synthesis, here's our cold DOTA MC1RL, and then after the radiochemical synthesis, we have actinium-225 in there. So this is our drug. So the radiochemical yield and purity, here are radio TLC chromatograms and HPLC, and we actually are doing our radiochemical synthesis at WashU in St. Louis, and they were able to get greater than 95% yield and greater than 99.7% purity, so that's really good. In vitro stability, in our 2019 paper, this was most of our preclinical studies were published in that paper. But you can see here, and this is in vitro, after 10 days, our drug is 90% still intact. So we have great in vitro stability. All right, so now here is the bulk of when I was involved in this, the preclinical IND enabling studies. So what you should do here is in vitro studies showing uptake, cytotoxicity, and radiobiology. And then you need to do in vivo studies with mice, or we did with mice. And you're going to look at toxicity, so MTD, mean maximum tolerated dose, kidney and liver function tests. We also looked at spleen and bone marrow and pathology. Then you look at the pharmacokinetics and the biodistribution, and then radiation dosimetry using the MERD organ level dose calculation method, and then efficacy in tumor-bearing mice. So I won't go into too much depth of these slides, but basically this is showing cytotoxicity and specificity of the drug in the starred cell lines, which are the mutations that are in metastatic uveal melanoma. So the lower on the green is better, so that's good. And then we have over here the specific and non-specific binding study. So this looks really nice, and our total binding looks pretty good. So then maximum tolerated dose and toxicity. We did a lot of work here. when I mentioned earlier where we tested several different linkers, we actually correlated those linkers with toxicities. I don't know if you can read it, but we looked at one of them is kidney, one of them is liver toxicity based on pathology, and then we looked at blood urea nitrogen, kidney function test, and then liver function test, and then weight loss. So our drug is in the black, and our different linkers are in the other colors. So the black is pretty good, or much better than the blue and the green. Then we also, in vivo, actually this was all in vivo, but we looked at weight gain. This is with our lead with the aminohexanoyl linker. We looked at the kidney function test. Then we looked at histopathology, and these are using our MTD determined by in the mouse, which was 4 microcuries. So we had a veterinarian pathologist look at the tissue specimens, and according to them, these are all healthy. So low toxicity there at our MTD. So then looking at the pharmacokinetics, we did some PK measurements in mice, and we determined, and then we also built a 5-compartment PK model. And we determined that the activity clears the blood compartment in about 20 minutes, so very rapid clearance. Biodistribution. Here again, on the left is the BD of different linkers, and if you can focus on the liver and the kidneys, you can see with the different linkers, you have quite different biodistribution. So when we're looking at just the lead that we went with, the aminohexanoyl, you can see that tumors expressing high MC1R have much greater uptake than tumors with low expressing MC1R, so specificity. And then here on the right is the BD for the gamma-emitting daughters that we can detect through gamma detection and tumor uptake. Then we do dosimetry. We use the whole organ MERD method, and there's the time activity curves that were fit with a mono-exponential. And then we get our dosimetry calculations. We did not use Olindo for this, but we did consult with Dr. Stabin, rest in peace, who helped us out. And you get all these PK, like effective half-life and things like this. So efficacy. So here are our representative images of basically control mice against the treated mouse, where after some time, the treated mouse pretty much has no tumor. And by the way, these mice were extremely sick and lethargic when we put these tumors in them. But after some time, the tumors were completely gone, and the mice were running around eating and drinking and lived out their normal lifespan as if they never had a tumor, which is pretty cool. So here we have tumor growth, tumor volume growth. So you can see our drug compared to the controls. Our drug is the purple, and we had significant growth delay of the tumor. Then we did some tumor burden analysis, and we saw that the treated pretty much got rid of compared to the saline. Green is the tumor. Same here. And then after treated, over here, we did some tissue analysis, and we saw that after treatment, the expression is generally the same as if it wasn't treated, which means we can probably start doing multi-dosing because the expression isn't gone. All right, summary. Running out of time. So, we had all these look good. So now it's time for FDA. So first in human phase 1 clinical trial. So in here, this is important, trial protocol preparation, IND, which is investigational new drug application preparation. You have the FDA pre-IND meeting, which you get to meet with the FDA and kind of get a live review of your application, and then you can go back and make your necessary changes and then submit the actual IND. So there's that. And then trial training and startup. This is your staff, the nuke med staff or whoever's involved, clinical trial coordinator. And then you initiate recruitment. So clinical trial protocol. Oh, I'm going to have to fly through this. So you have the study objectives. Primary, which is to assess the safety, tolerability, and determine maximum tolerated dose. And then secondary is evaluate the PK, the BD, the RD, and the efficacy. Then you have your study population, which includes the inclusion and exclusion criteria. And then you have your justification, which is kind of your evidence of your preclinical in vitro and in vivo work. Then you have your treatment plan. So you have to figure out your initial dose, your dose escalation scheme, DLT, dose-limiting toxicity definitions, and things like this. Then you have safety monitoring, PK and dosimetry, study procedures, statistical considerations. You need to team up with a biostatistician. And then regulatory and ethical compliance, which is in developing informed consent forms, IRB approval, and registration on clinicaltrials.gov. So for us, was the safety and tolerability of a single dose of our drug. And then secondary was the PK response again. Okay, so our inclusion criteria, I'll just say a couple of these. They have to be over 18. They have to have one prior line of therapy. They have to have measurable disease based on RECIST and acceptable organ and marrow function tests. Exclusion, they cannot have prior alpha therapy. They can't have a known CNS malignancy. They can't have any other concurrent type of treatment that could affect the study. So dose escalation. So our dose escalation is pretty neat. Okay. So we have 12 possible dose levels with escalation based on a modified Fibonacci sequence. So our dose escalation starts as the Fibonacci in the first couple levels where it doubles, but as you increase in levels, the level interval gets smaller. So it's a modified Fibonacci. Then we're also doing the CRM, which is the modified continual reassessment model. So it's adaptive dose finding design by continuously updating the dose toxicity curve based on data from previous patients. So the first in-human initial dose was based off of the no observed adverse effect level of the mouse, which we scaled to the average mass of a human. Then we divided this by 30 for safety. Then, as recommended by the FDA, we added 3 additional levels below this, this activity. So here's our dose escalation scheme. So our calculation on level 4, if we did this part right here and scaled it and then divided by 30, is 38. So this is kind of 4 microcuries was the mouse, and then based on some calculations, 38 ends up being for the human. And then we were advised to add 3 lower dose levels. So our first injected dose was almost the same activity as the mouse. Dose-limiting toxicity, DLT occurrence within 4 weeks after administration. Death would be a DLT. Neutropenia, thrombocytopenia, lab abnormalities, any grade 3 significant adverse event. So any of these, neutropenia and these, anything that's pretty much grade 3 or above would count as a DLT. So if the DLT occurs, you drop down to the previous dose level. And then if 6 patients are on a dose level that don't have a DLT, then the trial ends. So the maximum amount of patients we can have in the proposal was 30, and we've injected 13. So here's the schedule of activities. This is basically saying within 4 weeks prior to injection, they need to have, for example, consent, medical history, all these things need to be done within 4 weeks before injection. And then we take blood and urine for pharmacokinetics several days after, and then they come for follow-ups. IND application. IND application and requirements. Cover letter, qualifications of the investigators, the brochure, the CMC, the chemistry, manufacturing controls, the preliminary preclinical data, the trial design, which we just flew through, and commitments to obtain the IRB approval, informed consent from the research participants, and adherence to IND regulations. So here's our pipeline that we discussed. Upcoming developments, from that DoD grant that I mentioned, we have funding for the phase 1B multi-dosing trial of our drug. Then, in addition, in that DoD grant, we had funding for a phase 1 trial for a theranostic solution, gallium-67 SPECT. And then in the MRA grant, we got preclinical funding for the copper-64. And as I said earlier, 8 other cell surface targeted radiopharmaceutical therapies at various stages in the pipeline. So all of them are, I can't say too much, but all of them are at this level or beyond. So we've already identified targets for 8 other therapies. So here's just some acknowledgments. One of the benefits of working on this type of project is you get to have a very diverse group of experts to work with. And then it's also multi-institutional, which is very neat. So we worked with all these different institutions, and then here's our industry partners. And thank you.
Thank you, Chris. We don't have time for questions, so I would like to, at this time, introduce Corinne Smith. She is Associate Professor at UChan Medical Center, Department of Radiation Oncology. She is a Fellow of the AAPM, and she is the Director of Physics and Dosimetry at IROC, Rhode Island.
Thank you for the introduction. I do have a disclosure. My work is partially supported by the NCI through the IROC grant. And my perspective on this talk today is under the IROC umbrella. So I'll start with who is IROC, what do we do, and I'll talk about our core services that we currently provide for NCI-sponsored trials. And then we can talk about a vision for the future for services for our RPT trials in particular, pointing out differences and challenges, how they differ from existing radiotherapy and oncology imaging trials. So the history of IROC is that it was formed in 2014 at the same time that the NCI created the National Clinical Trials Network, or the NCTN. And under that, there were a lot of clinical trial groups that kind of got consolidated into 5 different clinical trial groups: Alliance, COG, ECOG Akron, NRG, and SWOG. And then we also do some support of the Canadian clinical trial group. But there were some incentives to share and collaborate between these groups. And if you've heard of RPC under MD Anderson, that became IROC, as well as the other offices that are under that umbrella. And an important part of this consolidation is that there was just going to be one quality assurance group to support these trials, and that is IROC. So we also support not just the NCTN, but other NCI efforts under consortiums. There is some clinical trials under the ETCTN that we also support as well. So our mission is to provide integrated radiation oncology and diagnostic imaging quality control programs in support of the NCI, in particular the National Clinical Trials Network, thereby assuring high-quality data for clinical trials that are designed to improve clinical outcomes for cancer patients worldwide. So these are our current core services for current NCTN trials. We have site qualification, trial design support and protocol development, RT credentialing and imaging qualification, data management, which is organizing and archiving data, case reviews, which is kind of a more radiotherapy-focused service where we do pre- and post-treatment review after radiotherapy. And I'll go into these a little more detail, and we can talk about how they might be applied to RPT trials as well. But this work is done to ensure high-quality protocol imaging and radiotherapy treatments and the collection, importantly, of high-quality clinical trial data. So I'll go in a little more depth about each of these core services and give some examples. So in protocol development, this is an imaging study. And in this step, IROC collaborates with the study committee, with the PIs on the study, and any medical physicists that are on the study as well, to ensure standard of care is written into the protocol, as well as if it's an imaging study, any advanced imaging parameters are clearly documented in the protocol. The protocol may also include standard of care imaging protocols, or with some locally determined variability in the acquisition parameters. Or the protocol may include some rigorous parameter specification and more involved imaging for an advanced imaging study. With site qualification, we're really trying to ensure that institutions can meet the basic qualifications for the protocol. The steps include making sure that enrolling institutions can submit data in required formats, and that they have the required systems. This is equipment and other systems involved. So enrolling institutions must complete a facility questionnaire to ensure that the site has the appropriate equipment and a team of individuals in place, so radiologists, radiation oncologists, research staff, et cetera, in order to support patients. Enrolling institutions must also conduct output checks for radiotherapy trials. The plot on the right here is a snapshot of the results of some of these output checks from linear accelerator checks. For proton trials, the enrolling institutions must go through a site approval process where someone actually comes on site and looks at your beam calibrations. Radiotherapy credentialing would be for radiotherapy trials in particular, and this is to ensure that the enrolling institution can meet the necessary qualifications for delivery of high-quality radiotherapy. So in addition to output checks on a linear accelerator, this is a more advanced credentialing test in the form of an end-to-end test, which may be in lung fandom as shown in the picture, or it could be proof of an image guidance technique on an existing patient. The goal is really to improve the understanding of the allowed techniques because the site ends up using the same protocol technique to deliver on a phantom, so it's a great sort of test of the whole process. And it's also to verify that the dose can be delivered accurately. So IROC will compare the dose, in vivo in the phantom compared to a predicted dose from the treatment planning system. And importantly, we work with medical physicists and dosimetrists to resolve any discrepancies that are found sometimes, but that can improve measurement results. And it's a really powerful step because it ultimately improves treatment delivery for all patients, not just protocol patients. So that's really important. And IROC, the Phantom Credentialing Program, has detected numerous TPS errors, treatment planning system errors, and these are documented in the literature and have been really informative for improving our models within treatment planning systems. For image qualification, if it's an oncology study with image submission required, just a quick note that this is a little bit different from accreditation requirements. Qualification is a prerequisite to site accrual, and the steps are just a little bit different than what would be required for your ACR accreditation. IROC case review is, again, specific for radiotherapy trials, but in this setting, we review the radiotherapy treatment plan, including the contours, the extent of disease from diagnostic imaging. We review the margins to ensure that they're all applied appropriately and as per the protocol. So IROC reviews the dosimetry in each treatment plan and compares it to the protocol parameters. In this example, this trial sought to de-escalate the dose in anus squamous cell carcinoma, and IROC conducted a pre-treatment review along with the study committee to ensure that the targets were appropriately defined since they were de-escalating the dose. And if not, if something needs to be revised, then we communicate back to the site. The site can submit, redo the plan, submit a revised plan that will then re-review. So this is also a really powerful step because it prevents protocol deviations, especially when we can do this review prior to the patient ever starting treatment. And preventing treatment deviations improves outcomes. So that's an important step. So IROC manages a massive amount of clinical trial data, and we have systems in place to connect to CTSU, which manages enrollment, Triad, where DICOM data can be submitted, in addition to other clinical trial databases like Metadata Rave, which is commonly used for clinical data collection. So with incoming data on enrolled subjects, IROC manages anonymization and encryption for privacy and security purposes, and we ensure that DICOM standards are followed. With this data, we can provide it to statisticians for primary analysis, and we can also, after the study is done and primary results are published, we can also provide this data for secondary research analysis. So how will IROC core services translate to RPT trials? And the short answer is that it's not quite clear yet. I think that there are still some challenges and some progress being made to get us to this point where we can have the same level of calibration and standardization as we do in our current trials. It kind of makes me think of when we were doing clinical trials before TG51, before we were calibrating our Linacs to within a certain degree of accuracy, before we were checking output on Linacs. We knew approximately how much dose patients were getting, but in order to really study the dose-effect relationship, we have to know to a great degree of accuracy, right? So I think that's where we're trying to get with RPT trials as well. So IROC has been working with experts in the field to educate ourselves. Many of you who've been giving great talks and in this meeting so that we can understand the needs for standardization and standard dosimetry within RPT trials. We have gotten a few talks from Stephen Graves and John Sunderland in our IROC research and development meetings. We attended the 2023 AAPM Summer School. Rob Hobbs, who's here, is a colleague, and he updates us on his efforts as well and his knowledge in this space. So we have a great group of experts who are, keeping us informed of the progress being made so that when we get to the point where we can start implementing some of this stuff, we're ready. This slide was shared with me by Rob. It's from the foundation of the National Institute of Health presentation to the ASTRO RPT Roundtable in April of this year. So PDIB is the Precision Dosimetry Imaging Biomarker Project. And this slide shows phase one of a two-phase project, which would essentially create the pathway for reliable dose calculations that will need to be able to study the dose-effect relationship. So on the right is sort of the the chain of things and how they need to go, going from the ground truth of a traceable radioactivity standard and all the way to calibrator, clinical radionuclide calibrator measurements, scanner calibration, patient-specific dosimetry, and then finally we can be able, with all of these systems in place and workflows, we can then be able to conduct large, randomized clinical trials where we can study the dose-effect relationship. So these efforts are all towards that goal. So these next slides are just sort of what would all these core services look like for an RPT trial once we get to that point, just trying to hypothesize a little bit. So site qualification would probably be pretty similar. The only challenge there is that these programs sometimes exist outside of normal radiotherapy and imaging departments. That's not a huge challenge. It would just be an extension of what we're doing currently to ensure that appropriate personnel are in place and equipment. Site qualification would likely also entail some verification of activity quantification. So institutional activity meters, should be NIST traceable for relevant sources, and we would likely, request some verification or upload of certificates in that regard. Another site qualification task may be to verify that institutions have the appropriate software in order to do patient-specific dosimetry. This is a list of software systems that are available. This came from Rob as well. And so there are many systems in place. The problem is that many do have FDA approval, but only for diagnostic or for post-treatment validation. So these systems are not quite approved for treatment planning yet. And then some of the older software systems, they give results in terms of effective dose and results in sieverts rather than gray, which would be needed to study the absorbed dose. So in trial design support and protocol development, this service would be little, similar to what we're doing now, where we would just provide guidance for accurate structure delineations from atomic images, recommend the dosimetry data reporting that is needed from sites enrolling patients, perhaps incorporate or facilitate radiobiology modeling to better predict biological outcome based on absorbed dose, identify elements of data collection in order to minimize the burden of data acquisition for sites, but also assuring that the necessary information is collected. And Ying Zhao at IROC Philadelphia has sort of outlined these steps that we could take for trial design support. In the process of RT credentialing and image qualification in this space, we'll probably have to come up with a new name. Doesn't quite fit into that bucket, but quantitative imaging, would likely come in as well as a step that we need to take and just verify that imaging systems can report the correct quantitative activity, including recovery coefficients. Graves and Sunderland noted that approximately 700 PET CT scanners have been validated over the past decade or so, that there are similar programs in place for SPECT as well, and that there are some phantom harmonization initiatives out there, albeit in their early phases of development, to establish standard phantoms and approaches, kind of as a corollary to what we're doing with credentialing in the RT space, external beam space. Data management would likely also look very similar for RTB trials. We would work with investigators to identify necessary data elements and from imaging that would be required to be submitted. We would assist in data submission. If everything were in DICOM format, that would be excellent, but we would work with software applications to come up with a standard format and make sure it's submitted. Identify and communicate what imaging and the number of time points that are necessary to collect, manage and communicate the protocol, site, and subject level dosimetry requirements, and then we could manage composite dosimetry as well. So we'd love to, do the case reviews as well, kind of similar to what we do for radiotherapy trials, where we can maybe review the dosimetry at each time point. We understand that there's a lot of reproducibility issues at this time, but ideally, we would look at the dose distributions, make sure that the calculations are based on activity. We could, validate the institution's dose calculation, perhaps on a reference case or a benchmark plan. And then IROC study teams would support the conduct of the trial by providing this real-time review of quantitative imaging, performing organ delineation segmentation as needed, image registration, and extracting the radiation oncology objects that are of importance for the study. So, IROC ensures that the patient is enrolled in the correct study and that all the appropriate data, including the imaging time points and any radiotherapy objects, are submitted. And then importantly, we communicate feedback back to investigators in a timely manner. So in addition to the challenges that we've sort of already kind of reviewed, there's additional challenges of just the cost and effort associated with nuclear imaging examinations, not necessarily covered by insurance at the moment, but they are necessary for the processes that we're talking about and for dose calculation, image registration, and to be able to study the dose effect relationship. And the lack of availability of standard treatment planning methods and software systems. But we are here for the task, and we will perform, all of these core services for any NCTN RPT trial that comes along. So our infrastructure and our service operations, our support will be made available to participating institutions as well as study committees. And this approach, like other trials, enables centralized quality assurance, centralized reconstruction, independent validation, and standardized image analysis and dosimetry methodologies. And that's it. Thank you for your time.
Thank you, Corinne. That's very exciting. Do we have any questions? We have literally 1.5 minutes. No? Well, I was curious. Oh, sorry, very good.
Yeah.
Because we're undergoing a number of different collecting of data, and we've been having trouble finding places where we can store things and anonymize things. So are you open to data storage outside of NCTN trials, or does it have to be within the, because like just within the